I'm the managing partner of Transcript Partners, Flick Gabay, and I am going to introduce Chris Brearley to you because this in our series of clinical development videos covers early development all the way from late stage preclinical to proof of concept, including the first in man studies. Now, first off, I'm going to introduce Chris. He's a pharmaceutical physician of 20 years standing. He was originally qualified in nephrology and clinical pharmacology. And during his 20 years in Big Pharma, he has studied small molecules and biologicals, so has a considerable amount of experience. He uses that experience in Transcript to be head of our translational team, and we have a number of senior pharmaceutical physicians who have many years' experience of early development. So Chris, the target of first in man, that big jump into humans after early clinical development, is a big milestone for companies and often a financial milestone as well.
This is a very difficult time for small companies which have many drivers, particularly financial drivers at this stage. One of the key things in early phase drug development is planning for success and ensuring that one can maximise the value of the asset during the early development programme. The early development programme is not just a series of sequential studies, a SAD followed by a MAD, followed by perhaps a food effects study, and then entry into patients. One should really design these programmes by deciding what the best proof of concept for this new novel target is going to be. Having decided upon the proof of concept, how are we going to demonstrate clinically meaningful activity of our new drug in man? We can then go and design the journey to achieving that critical goal.
Okay, so it's not just about increasing the dose in single dose studies till you get to maximum tolerated dose, and then looking at tolerated doses in multiple dose. We can learn a lot more, can we?
Yes, Flick. I think one of the key things about the early phase programme is by the time one goes into the design of the proof of concept study, you want to have a clear idea of the effect the body is having on your drug, i.e. the drug's pharmacokinetics, and the effect the drug is having on the body, i.e. the body's pharmacodynamic response. Clearly, there are always assays to measure the drug itself or its metabolites, but it's important to carefully select biomarkers so that one can assess the pharmacodynamic effect of the drug on the body. And again, when designing the early phase development program, it's here that starting with the design of your proof of concept study and what your objectives of this phase are, lead you to which biomarkers should we be measuring and what are the most important biomarkers to measure. Biomarkers can be biomarkers of drug effect, i.e. the pharmacological effects of the drug, or they may be a consequence of that pharmacological effect, i.e. a change in heart rate, a change in airways responsiveness, as perhaps measured by forced expiratory volumes.
I guess that means that you would be then preparing to get the right dose for proof of concept. I'm very conscious of the fact that when I pick these things up in phase 2 and phase 3, one of the commonest issues is that we get the dose wrong.
Indeed, by paying careful attention to concentration effect and concentration adverse effect relationships in the early phase program, in designing the proof of concept study, you're selecting doses that you understand and you know are going to have a meaningful effect that you want to see translate into a clinically meaningful effect in the proof of concept study. And by understanding these concentration effect and adverse effect relationships well, you minimise the risks to patients and healthy volunteers.
So summing up, what am I going to learn out of early development that I will be using in my POC phase 2 and subsequently my clinical studies in phase 3?
From the data generated with carefully and rationally selected safety and efficacy biomarkers, we are going to be able to demonstrate drug activity, may be able to differentiate the drug from other competitive molecules that may be in development or already in the marketplace. We will have defined our safety liabilities, and we will also have demonstrated a dose adverse effect relationship and a dose response relationship, which we can use for rational dose selection in designing the proof of concept study. Hence, we're building success and safety into the proof of concept study.